Un, deux, un, deux, un, deux, un, deux, un, deux, un, deux. On est bon.
Ça commence à aller mieux. Tu penses que non, mais là, ça fonctionne, c'est bon.
Un, deux, un, deux, un, deux, un, deux.
Je vous laisse tester si le son vous convient, si c'est trop fort, pas assez fort. Selon comment vous parlez.
Est-ce que ça va comme ça? Oui, je pense. J'ai pas une voix qui parte hyper fort, mais la salle n'est pas immense non plus. Là, c'est bon? C'est bon? Hop.
Bonjour.
Un, deux, un, deux. Un, deux. Un, deux, un, deux. Un, deux. Un, deux, un, deux, deux. Un, deux, un, deux.
Yes, can you hear me? Can you hear me well? So just test, audio test. Can you hear me? Test. Okay. Yes. Yeah, it's working perfectly well. Okay. Thank you very much. Then I'll connect at 10 past 9. Is it okay? Or even before, I can connect at 9 as you wish. I'm here all the time, so it's no problem. Okay. Then let's do that. Yeah, no problem. It's fine. Okay. Thanks a lot. Thank you very much. Sharing.
m'entends bien? Oh, ok.
Oui. Oui, je pense qu'on m'entend bien. Allez.
Ah ouais, c'est ça. Bah, il est 8h31. Attends, 3 minutes.
Ah bah il y a tout le monde qui arrive. 30 secondes encore? Ah, d'accord. Ok. Merci. Okay. On va rappeler que dans le ouais. Ouais, ça marche. Il va pas vraiment laisser Karine en fait. So, just before starting, we have an announcement. People need to remove their poster. I think it's for the party tonight, so better to do it. C'est très désagréable, c'est... Ouais. C'est bon? On peut commencer? Non, c'est bon? Okay. So, good morning everyone. So I will introduce the first speaker of this last morning session of the MIFOBIO, so Charlotte Rivière. She's an assistant professor in Lyon, and she's working at the Institut Lumière Matière, also in Lyon. And she managed to combine biophysics, cell biology, microfluidics, microscopy, and image analysis to study fundamental questions in mechanobiology, or more apply project in cancer biology. I think it will be the topic of today. And she also recently received CNRS bronze medal for her work, and that also helped me to prepare the introduction. So, I mean, I was not the one who invited you, but thanks a lot for coming, and I let you present your work.
So thank you very much for the kind introductions, and thank you very much to have the possibility to present our work here. It's an amazing congress workshop, and so I'm really happy, and thank you for all of you who have managed to wake up at early morning. So I'm Charlotte Rivière, and so I will talk about what I will talk about today. It's how we can quantify transport of nanotherapeutics using a spheroid model, mainly. So indeed, as Stéphanie Decroix and Laurent Malaquin also stated yesterday, most of drugs are still largely flayed when they go up to phase 2 and phase 3, and especially in oncology. So you've got here the success rate. In oncology, it's only 5% of the drugs. Maybe it goes in this one. In oncology, it's only 5% of the drugs that go from phase 1 to phase 2. And there is many reasons for that, but one reason is that there is a huge gap in between the 2D in vitro models and the in vivo models. And to fill that gap, there are many possibilities to try to replicate in a more or less mimic environment, the tumor microenvironment. So there is a different approach to do that. What we are doing in our lab is to use spheroid model, so this kind of models. So indeed, we are not flat, and compared to 2D cell cultures, there is lots of things that is missing in 2D cell cultures. Cells in vivo will be submitted to different type of interactions, cell-cell interactions, but also cell matrix interactions, biochemical stimuli, and also mechanical stimuli. I will talk a little bit at the end of my talk about mechanical stimuli, because we are also interested in mechanobiology in our lab. So indeed, so what we are doing, we are using spheroids. So spheroids is a kind of the simplest 3D cell models. Why? Because we can reproduce the complexity in terms of different type of cells within a tumor, and it's still possible to make some models out of it compared to more complex ones. So you can, for those who are not aware of spheroids, you can, if you have large spheroids, you can have necrotic cores. Then in the middle, you have some cells that are not proliferating, which are called quiescent cells. And on the periphery, you have the proliferated cells. So the cells that are proliferating are only in the periphery. And this is kind of similar to what we can find for a microtumor in vivo. And so I will talk mainly about nanoparticles transport, and for as long as nanoparticles are concerned, the main issue is to be able to go from the bloodstreams, which are produced here, to the core of the tumor, because this is a very dense medium. And so it has been shown that this is one possible models to analyze the drug transport of nanoparticles within this kind of 3D model. So how to make spheroids? The classical way to make spheroids is this one. So it's ultra low adhesion plate. You've got a U shape here, so all the cells will be sedimented at the end of the bottom of these wells, and if they have cell-cell adhesions, proteins such as cadherins, they will self-assembled into spheroids. And so this is a couple of hours, but then it will be more and more compact, and after a few days you will end up with something like that. So to do that means that you need to do 96 pipetting and 96 to get 96 spheroids. And if you want to image your spheroids, if you want to do immunostaining, that means that you need to do lots of handling process that can be very long, very difficult, and you can damage your spheroid. And so what we try to do now is to try to combine this spheroids approach with this ultra-low adhesion plate with microfluidics. So this has been nicely introduced yesterday already. Microfluidics is a way to make everything in parallel at a very small scale. So you can have different drug concentration in parallel, you can have different therapeutic treatments. We are also interested in different mechanical stimulation. So the idea is to combine these 3D models with microfluidics to have a more parallelized high throughputs. So there are many people that have tried to do that. So this is 2 examples of combination of microfluidics for spheroids. So in this one, you've got here an array of microchambers that will lead the cells. I don't know if you see. I will try to see. You have got one single cell here at the beginning, and 28 days after, then you have your spheroids. So this is a very tricky possibility. And another possibility is that you're creating your organoid before, and then you inject your organoid and you trap them. So you've got some kind of trapping systems here to trap every organoid, so that it's possible to image and to have a high throughput on the analysis of these organoids, but you made them before. So in this kind of setup, the polymer that is used to make these microfluidic systems is PDMS. For those who don't know this approach, so the idea is kind of simple. So you have your design, and you are using SU8 photoresist resins to make this design in 3D, and then you pour your PDMS. It's very viscous, and then you bake it, and you end up with, and you make some holes, and you end up with this kind of systems. For example, here you have multiple chambers. The protocols are very well known. It's easy to do when you are formed, and it's transparent, so it's good for microscopy. But there is some issue that needs to be solved, especially for drug screening, because hydrophobic molecules tend to absorb on the surface of the PDMS. And so if you want to do drug screening, it's an important issue, because the concentrations that you will have in your systems will be very different, very low, compared to the concentrations that you expected. And the second point also is that the PDMS is also still kind of rigid. It's a megapascal range, while in vivo it's in the kilopascal range. So it's orders of magnitude higher. And also it's impermeable to small molecules. So that means, and here the volume is very, very low. It's in the nanoliter range. So that means that if you put your cells in such a medium, very rapidly your medium will be conditioned by your cells. They will secrete some stuff. They will get some nutriments. So you need to make some renewal in somehow of this medium if you want to make PDMS. And so this is more complicated first, and the second is that you can then your cells will also be submitted to shear stress. So to try to cope with this kind of issue, there are lots of people that are making hydrogel-based microsystems. I've chosen 2 examples from the same team, actually, from Matthias Luthoff in EPFL in Switzerland. So this one was an early one in 2014. So here you've got so everything is in hydrogels, and you can put here only 2D cells or spheroids culture, and underneath you have a system that is connected to a fluidic system, so you can make a gradient of molecules, and you can analyze the gradient of how cells will respond to the gradient of molecules. And this is from the same teams, but very recently. It's a Nature paper in 2020. It's a very elegant systems where they combine PDMS with hydrogel. So in the middle chamber here, you can have hydrogel. It's a mix of matrigel and collagen. And they were able to recreate a microvilli. So this is what you have here, this microvilli. And in the middle, they are able to perfuse this organoid on a chip with medium. So they managed to maintain this microvilli for several weeks, compared to what this cannot be achieved without this microvilli setups, and you need to break again your organoids and put them back. And here they can maintain this. So this is really super cool stuff, but it's kind of complicated to have it as a routine system in a biological lab. So we went to a different approach, much less complicated, and we are developing in the lab microsystems in agarose. So why agarose? Because it's very cheap, and also it do not swell compared to most hydrogels. So as it do not swell, that means that you can get your design is really the dimension of your design. You can keep it when you do the molding of the agarose. It's cell repellent, so that means that the cells, as in ultra low adhesion plate, will not adhere on the agarose. and there is no known biomolecular absorptions. And also it's closer to physiological rigidity. You can tune the rigidity of the agarose depending of the type and concentration between 1 and 100 kilopascal. So it's much closer to the rigidity. So this is how it looks. So here you've got a cover slip and an array of microwells. So the design we are using now is microwells of 200 micrometers most of the times, because we want to get small spheroids 200 micrometers that are that is easier to see with microscopy. And in these 200 micrometer spheroids, you have only two kind of layers. You have the proliferating cells around and the quiescent cells, but you do not have a necrotic core. So if you compare to the like classical ultra-low adhesion plate, here you get 96 spheroids for 96 pipetting. In our case, most of the time we are using 24 multi-well plate, and in each plate you can have this microwells array in agarose with more than 100 spheroids, so you can do more than 3,000 spheroids in only 24 pipetting, and in parallel you can have different conditions within the same plate. And you can handle it as if you are handling a classical 24 well plate, so you can change the medium very easily, so you can do every step of the immunostaining in situ without moving your spheroids. You can move your agarose plate, but you do not move your spheroids, so you keep them intact. And also you can perform optical imaging. So for to be sure to have maximum oxynal, usually you can put our microwells upside down so that we are very closer to the objectives, and we can do Z-stack in multi-positions with these spheroids. So to show you some examples, so this is your low magnification. So all the spheroids are almost in the same focus plane, so it's easy to do multi-position and to analyze, to have high statistics on this kind of setup. So using these systems, I will present two different things. First of all, I will present something that has been published recently in Labonship on the use of these systems to analyze transport of nanoparticles. And then I will present you some unpublished work on the effect on radiotherapy and electrochemotherapy. And if I have time, I will just present briefly the role of mechanical strain on spheroids, because we are also interested in this in our team. So concerning the first one on nanoparticle transport. Oops. Sorry for it's not my computer. I didn't check that. So here, this is the work of Saba Goudarzi, a brilliant PhD student that has been her defense was in last month, and we co-supervise with François Luce, who is also from the Institute of Light and Matter in the chemistry part, and he has developed nice nanoparticles. I will just present now. So these particles that Saba and I used are called Agix nanoparticles. They are made of gadolinium and they are surrounded by a polysiloxane matrix. They are in phase 2 clinical trials with this company, NH Agix. They are used as MRI contrast agent, and as they are very small, they will also passively accumulate within tumors. So here you've got many metastases in the brain, and this is the signal of the Agix. So they are accumulate passively in the tumors, and they are radiosensitive. And so the idea is to combine these nanoparticles with other type of therapy to improve the therapy. So in our case, we want to analyze the transport using optical imaging, so they combine this with an optical dye, C5, to be able to see them with optical imaging. So first of all, we analyze globally the concentration of nanoparticles within our spheroids. So we are using HET 116 spheroids, which are colorectal cancer cell line, and 200 micrometer spheroids. And so this is for different concentration of nanoparticles in 2D in white and in 3D in spheroids in black, the total amount of nanoparticles that are found in the different system. And you can see that for low concentration you have almost the same amount, but when you go to higher concentrations, you have a larger difference between 2D and 3D. So this is easy to understand, because in 2D every cells will have access to the same batch concentrations, while in 3D the cell in the periphery will have more concentrations than the cell in the center. So this is only a global analysis, an average analysis per spheroid, but then we wanted to go to in situ analysis to analyze as a function of the distance from the periphery what is going on. So for this we performed Z-stack analysis using confocal imaging, and as most of you know, in confocal imaging and optical microscopy, these kind of spheroids are very opaque. Because there is some kind of mismatch of refractive index. So to be able to go up to the center, even for these 200 micrometer spheroids, we need to use clarification technique to make them transparent. What we are using, as they are very small, it's 200 micrometers, we do not need to use very complicated clarification technique. Glycerol is working very well, so this is what we are using with this kind of spheroid. So you've got an example here of a controlled spheroid without clarification, almost in the middle plane of the spheroid. So you can see that in the border you see the cells, but in the middle it's kind of black. While when we clear them overnight with glycerol, we clearly see the nucleus. So here it's only the nuclei, just to show you how it looks. So we've done that with our nanoparticles. So here in red, it's in our nanoparticles for different time after incubation and for different depths. So for 1 hour, we barely have any accumulation of nanoparticles, and they go almost up to the center at 24 hours and 72 hours. So out of this image, we spent a lot of time to try to quantify it as a function of the distance from the periphery. And so this is the kind of graph we can do. So here we've got the mean intensity as a function of the distance from the center. So here at 0, you are at the periphery. So this is where you have the highest concentration of nanoparticles. And here you are in the center of the spheroid. And the bolded line is the average over several 50 or up to 100 spheroids. For 1 hour, you have low concentrations, but you can see that you go up to the centers for 24 hours. So in most of the experiment afterwards, I will show you, we keep these 24 hours because the nanoparticles manage to get up to the center. And so now we wanted to know, as when we knew that it was possible for the nanoparticles to go up to the center, we wanted to know what was the internalization pathway. And so for that, we first performed immunostaining with different organelles. So this is a result of immunostaining in 2D and in 3D. So here for different organelles, early endosome and lysosome. So we also have done it for mitochondria. In blue, you've got the nuclei. In red, you've got the IX, and you've got either lysosome, either early endosome in green, so that the co-localization will appear in yellow in these images. And you can see here qualitatively that most nanoparticles, the yellow points here are mostly in the lysosomes for both 2D and 3D. But it's not really quantitative to have this kind of image. So to make and to analyze what's going on as a function of the distance from the periphery, we work on a way to quantify the colocalization. So what we used the Pearson correlation coefficient, which is a classical method for for colocalization. So for those who don't know, the idea is to analyze for every pixels in the different channels, the red channel and the green channel, the difference between the average intensity, and to multiply these two. So when you have, and to normalize, and so when you have this Pearson correlation coefficient that is close to 1, that means that you have a perfect colocalization. And when it's close to 0, it's not colocalized at all. And the threshold we use usually is 0.5 to say that at least it's partially colocalized. So we've done this in 3D in our Z stack. And this is the result for the moment only on the average in 2D for both 2D cell culture and 3D spheroids. So every dot here, it's a plane. And so you've got different spheroids or different 2D cell cultures and different planes. And you've got these percent correlation coefficients for early endosome, mitochondria, and lysosomes. So first of all, you see that we see that early lysosomes is the main organelles where we found nanoparticles, but it's different in 2D and in 3D concerning early endosome and mitochondria. So in 2D, you get a lot of nanoparticles that are still in early endosomes and very few in mitochondria, while it's the opposite in 3D. So we didn't go deeper than that for the moment, but it will be interesting to know why. The idea is that, again, the nanoparticles access is really different in between 2D and 3D, and it has been shown that the nanoparticle internalizations will depend on the concentration, local concentrations, and as the local concentration is different in 2D and in 3D, this will enable maybe to go to different routes and different pathways. So, as we know now that nanoparticles are reaching the center of the spheroids, they are mainly within the lysosomes. The second thing was to know the analysis of the radiotherapy, because our nanoparticles are radiosensitive elements. So for this, what we have done, and we have compared again 2D and 3D. So we have performed our irradiation with a range of different irradiation, and we have dissociated both single cells, 2D cells, and spheroids to perform the gold standard in radiotherapy, which is the clonogenic survival assay. So what they are doing is that they are diluting the cells at maximum, so that they have single cells, and they are analyzing the cells that are able to make at least 6 divisions. So this is a gold standard, and so they are counting the colony, and out of this colony they are estimating the survival fraction. So this is what we have done. So this is the survival fraction as a function of the irradiated dose for 2D in triangles, and for 3D because it's a circles. So first of all, for large gray value, 6 gray, we do not have points for 2D because all cells, no cells are surviving. So it's really different in 2D and in 3D. The survival rate is really different. And if we compare for the lower gray value at 2 gray, you've got almost twice most cells that are surviving in 3D. So you've got here 30% of the cells that are surviving, and in 2D it's only 12%. So the survival fraction is really different in 2D and in 3D, and you have a higher radio resistance in 3D. So that's why it's very important to, for this analysis, to keep the 3D structures. We thought it was even important also not to dissociate the spheroids, to analyze what's going on in situ within a 3D context. So we try to do that, to keep the spheroids within their 3D context. So this is what we have done. So we seed the spheroids, we incubate with the nanoparticles, and then after the irradiations, we keep them and we follow their growth for several days. And so this is possible thanks to our microsystems. We can follow with time-lapse multiple positions and quite easily. And then from this, so this is what Saba has done for different conditions. So it's a bit a nightmare for segmentations because, so finally we ended with manual segmentations. Maybe there is a clever way, but Saba spent a lot of time to analyze for both conditions. So I just put here 2, the control, and the 2 gray. So here you've got the volume as a function of time. Without a Gix, this is a gray point, and with a Gix with nanoparticles, this is a pink point. And 1 point here is a single spheroid, and you've got the average in black or in red. So we were quite disappointing because, as you see, well, there is no difference. So the first thing is that the spheroids are still able to grow even at 2 gray, but there is not a significant difference between the spheroid growth with agics or without agics. But maybe this is only the global average here, what you see, the global growth. So, but maybe there is a really different in situ. So we wanted to go to in situ imaging to analyze if there is a specific population that are still surviving within our spheroids. So for this, we have done immunostaining to analyze proliferative cells. So we incorporate EDU at 9 days after formation of the spheroids, and then we are performing this kind of imaging, so 3D stacks again in EDU. So here it will appear, the colocalized cells, so the cells that are proliferating, will appear in yellow. So I don't know if you've seen, but most of the cells that appeared in yellow in this kind of 3D stacks are in the periphery here. So here it's maybe easier to see. So you've got for the control at different depths and in the presence of agics nanoparticles. So first of all, even if we clarify them, we lose some signals here, but it's not zero. We still have some signal, but it's a bit lower because they are very dense at 10 days. And so what we perform, we used again this Pearson correlation coefficient, and we average as a function of the distance from the periphery the Pearson correlation coefficient, so the colocalization between the nuclei and the EDU staining. We took the threshold of 0.5, so the cells, the layers that have a Pearson correlation coefficient larger than 0.5, we took them as proliferative cells. And if we do that for control in green here and for nanoparticles, agics nanoparticles in red, you can estimate a proliferative layer, and you see that it's kind of different. So you've got a proliferative layer of 60 micrometers almost for control cells, and it goes down to 30 micrometers when in the presence of nanoparticles. So we are affecting the proliferative layers in our spheroids with these nanoparticles. So we have a higher radiosensitive effect, even if we do not see it with global proliferation growth. And so this is what we are now concerning the radiotherapy. What we want to do next is to analyze other properties and how radiotherapy will affect other properties of the cells, and in particular how it will affect invasion properties. So this is just a preliminary experiment. In our microsystems, as we are agarose, we can revert them and put them in a layer of collagen, for example, and then if they have some invasive properties, they can go out and invade. So from this movie here, the spheroids were too close. It's difficult to get any quantitative analysis of this, but this is the idea. We want then to have spheroids that are more apart to be able to do some quantitative analysis out of that. And also what we want to analyze if the influence of other factor of the tumor microenvironment, so the presence of the matrix, the presence of cancer-associated fibroblasts, and also we want to go towards larger spheroids where we have a necrotic core and hypoxia, because hypoxia is an important factor that is going to radioresistance, and so this is where we want to go now. So this was the first chemotherapy application. And I will present more briefly a second one, which is electrochemotherapy. So this is a collaboration work with Ampere team, so the team of Marie Robin, and we are co-supervising Pauline Bréquigeon, which is in second year. So they are working on electrochemotherapy. So what is electrochemotherapy? So it is the possibility to incorporate some drugs within cells by temporally permeabilizing the membrane. So this is the idea. You've got a molecule that will not enter into the cells if the cell is intact, if the membrane of the cell is intact, but if you temporarily permeabilize it, you can incorporate your molecules. And there is two types of electroperforation applications. Either you put some drugs, and this is called electrochemotherapy, and you can also put some DNA, and this is called electrogen therapy. So electrochemotherapy, it's already in the clinic. It's used in the clinic, but it's difficult again to be able to optimize what are the conditions of the electroporations in a 3D context. And so this is why Marie came to me to see if it was possible to use our microsystems to build 3D models to better understand and optimize electroporation. So what they are doing to make electroporation for 3D spheroids, there is no way to do it commercially. The commercial electroporators are made basically for single cells, and if you do that for spheroids, there is lots of hanging steps, and also your spheroids can be electroporated very differently if they are close to the surface of an electrode or in the middle. So it's not very handy for spheroids. So there are some people that have made some microsystems to try to do that for spheroids, but it's still complicated. And so the idea was to combine our microwave within a microfluidic chip to be able to do everything in situ. So the spheroid growth, with precise spacing, so they can control the electrochemotherapy, and they can exchange the medium quite easily because of this microfluidics. And we can do that, and we can do analysis with optical imaging, so we can do in situ high content screening within these microsystems. So first of all, she analyzed it. The global analysis of this electroporation with live and day approach. So here, if your cells are dead, they incorporate PI. But if they are alive and electroporated, you will still have PI, and so you will have both the red channels and the green channels, and they will appear green. So they are alive, but they are unincorporated PI. So we're using PI as a marker of electroporation. And so you can see here from these images that so she increased the voltage at only at 200 volts, that you have almost all your spheroids. So here all the dots are single spheroids. It's a large magnifications, and all the spheroids are yellow. And so she quantified that, and you've got a real difference in the mean red fluorescence between the 100 volt and 200 volt. And the other thing that we need to check is if it's really reversible. So this is what she checked 2 hours later. So 2 hours later, the membrane is back again, is not permeabilized anymore. So PI should not enter into the membrane. And so this is what she checked. You have almost no dead cells, and it's reversible electroporation. So this was the proof of concept that it was possible to electroporate thousands of spheroids within the same setup. And so here it was time to try to know if electrochemotherapy was possible. But before, we wanted, so this is a global analysis for every spheroid, but we wanted to know in situ if all the cells of the spheroids were electroporated the same way. So for this, we have done confocal imaging, Z-stack confocal imaging. I don't know if the contrast is very good, but so here you've got one plane almost at the center of the spheroid, and here it's the Z projection. For low voltage, and even at 200 volts, that was our threshold before, you can see that in the periphery you have PI, but in the center you still do not have PI. So that means that even if from global analysis we thought that 200 volt was enough, in fact, if you want to electroporate all the cells, we need to go to higher voltage, and in 300 volt we managed to electroporate all the cells. And so knowing that, she has done electroporation using a drug that is bleomycin. So bleomycin will not enter the cells if there is no electroporation. So that means that if you put your spheroid in contact with bleomycin only or electroporation only, the spheroids will continue to grow. And it's only if you electroporate, that bleomycins will enter the cell and will have this therapeutic efficacy. So this is what she quantified, and this is the images. So here you got the evolution of the spheroid size as a function of time, up to 3 days. For the different control conditions, the spheroids are growing, and when in the presence of bleomycin and electroporation, there is almost no spheroid growth. So in this system, it's easy to. She managed to make a microfluidic chip that is possible, where it is possible to analyze and to monitor several spheroids in parallel. So it's interesting now to analyze what's the effect of the electric field, different shape of electric field, how it will be influenced with the size of the spheroids. Sorry. And again, we have the same question now: what is the influence of the matrix? What is the influence of the cancer fibroblast? And how the necrotic core, the presence of a necrotic core, will influence the electroporation. So to finish, I just want to briefly show you the effect of mechanical stress on spheroids. So to come back on the first slide of my presentation, I told you that there is also mechanical stimuli within in the tumor microenvironment. So the cells are submitted to different in stiffness. They will grow within a defined volume, and so they will be compressed at some way. And if they are close to the blood flow, they will also be submitted to shear stress. So to try to understand what is the role of this mechanical stimuli, there's different way to try to compress cells. So this is, you can put them in some kind of channels, open channels where your spheroid is growing, but it does not have too much space. Here you can use the capsule, alginate capsule here. Again, they will need to push on the capsule. And you can also use agarose and dextran, which is a polymer. If you take a large dextran, it will not enter into the spheroids, and so it will exert some kind of pressure, also osmotic pressure. So in our lab, we are using these two kind of possibilities, so either agarose, either dextran. And what I will show you today is both. So first of all, in a previous PhD thesis of Audrey Prunet that I co-supervised with Hélène, we used, so I told you at the beginning that it was possible to tune the rigidity of the agarose, and we can go up to very low rigidity, 2 kilopascals, so it's close to in vivo rigidity. And if we do that, and we can also include some beads. So I don't know if you see here, you've got small, it's fluorescent, but you have small beads, and this is a growing spheroids that will push on the wall, because here the agarose wall are very soft, so the spheroids, when it's growing, it's possible for him to push on the wall. And so from this kind of image, we can track these small nanoparticles, and we can, from this, as we know the elastic properties of the agarose, we can analyze the deformation fields and the traction force microscopy. I will just show you the deformation fields. What we saw is that, in fact, we do not have something that is isotropic. It's only very local events. So the cells are growing, and at some point they will push, and there is some space, and they will continue to grow. So this is one possibility, but in fact, here the compressed stress is not building very fast, because it's possible, as it is possible for the spheroids to grow, the compressed stress will stay low most of the time. To include some more some mechanical stress, higher mechanical stress, one possibility is to use this dextran. So this has been performed with the team in MSA 10 years ago. So here you've got, so it's spheroids without pressure and with 1 kilopascal pressure. In the center here, you do not have any more proliferating cells. So the cells the mechanical stress will impair cell proliferation. And this has been also been shown more recently, where you've got here the evolution of the radius as a function of time for a control spheroid and a spheroid with a chemotherapeutic agent in yellow here. And in the presence of this stress, we impair the chemotherapeutic efficacy. So the purple curve here, it's a bit above the yellow one. So that means that the chemotherapeutic agents will also be affected, the chemotherapeutic properties will be affected by the fact that the mechanical stress will induce some modification of the cells. Most of the therapeutic agents are targeting proliferating cells, and as mechanical stress will impair cell proliferation, it will also impair the chemotherapeutic therapy. What we are interested in the lab is the role of mechanical stress in cancer stem-like cells. So I don't know if you're aware of these cells. So it's a specific subpopulation of cells which are called cancer stem-like because they have some stem cell properties. And the hypothesis is that even if you manage to kill almost every cell, there is these cancer stem cells that are remaining and that will be responsible for the relapse. So to understand this really subpopulation of cells is very important to analyze what's going on. And so for this, this is part of the PhD work of Malek Moueli, who is here, and co-supervised with Sylvain Monnier from our lab. So we combine the dextran with our agarose microsystems because then we can have a large statistic very easily. And what I will show you is the evolution of the nanog expressions, which is one of the stemness marker as a function of time, here for a control and here under the pressure. So it's epifluorescence, and it's in false color. So in high, it will be in white. And if you have a high expression of nanog, and if you have a low expression of nanog, it will be in blue. And so this is the movie. So I don't know if you see clearly, but you have a large expression of nanog within the pressured spheroids, while in the control, it's much lower. So Malek quantified this with different pressure, and so you've got the normalized area as a function of time. So and here the intensity, fluorescence intensity as a function of time, and we have in the same time a decrease of the spheroid growth upon stress, and in the same time you've got an increase of this nanog expression. So that means that it might be some increase of stemness capacity we can induce by the mechanical stress. So we show that we have, using this colorectal cancer cell line 1829, we had an impaired spheroid growth and an increase in nanog expressions. So is it really favoring stemness taste? We still do not know, and how it will impact resistance to therapeutics. This is an open question. So this is what we want to do next. We want to analyze other stemness markers and also to perform Western blot and qPCR to really know if the protein expression is increasing upon mechanical stress. And also, again, we want to analyze in situ, because here it was only a global analysis, and we want to know if this is uniform within the spheroids or if there is only a specific spots within the spheroids that express these stemness markers. And just to finish, so I've shown you agarose-based microsystems that can be used to analyze in situ nanoparticle transport and response to radiotherapy. We can have both global growth follow-up, but also in situ single-cell analysis, and it's compatible with many types of stimulation, radiotherapy, electrochemontherapy, or mechanical stress, and also we can combine some different type of therapy. And so I'm really turning with application to cancer cell biology and drug screening, and we'll have some new experimental lab in the Research Center for Cancer. So I will just show you one slide left for to show you that our system is also compatible with patient-derived cells. So this is a project with the team of Philippe Bertolino and Audrey Ziverek. And this is a specific type of tumor which are called gonadotroph tumor. And what they are doing is that from patient biopsy of patients, they are putting cells from these patients in our microsystems, and they want to analyze different biomarkers, and they want to analyze the tumor heterogeneity within our microsystems. So with this, I want to thank you for your attention, and I want to acknowledge all the people I'm working with and the funding, so the biophysics team, the Fennec teams from the ELM. We have also a large engineering support from our institute. I didn't show it, but we also have a collaboration with David Rochaud from Angers and Gaël concerning the segmentation of the spheroid in situ, so Ampere and CRCL, and EMP Arnaud Favier with another kind of nanoparticles. And I want to thank you for your attention.
Okay. Yes. So, question.
Thank you. Thank you for your presentation. I have a question about the electroporation. When you do the electroporation of the spheroids or organoids, how resistant are they? Because in my experience, if I do electroporation of normal eukaryotic cells, there is a still part of them which will die just because of the electroporation process. So how many of the spheroids will survive so you can use them later for the therapy?
Actually, the rate of dead cells are really low. It's below 3%. So this was quantified. If you perform, maybe I will just come back on the slide. But if you perform electroporation with the classical system, usually it's really not optimized in terms of electrodes and voltage, and so maybe you have a large death because there are maybe some of them are really close to the electrodes and some are not. And here, the fact that they are really within the same plane, she can optimize electrochemotherapy, the electroporation, so that you have a limited depth. So we quantify that it's below 3%. So almost all of them are.
Thank you very much. Do you have any insight on the extracellular space in your spheroids? Do they grow extracellular matrix, and do you have different textures?
This is an interesting question. This is we want to analyze that, but we didn't yet. The model we were using so far, HET116, it's known that it's not making a lot of matrix, but maybe there is some. And what we want to do now is to combine that in the presence of fibroblasts that will create some matrix to see what's the difference. But yeah, that's an interesting point.
Hi, great talk. So I just had a question about the drug penetration with the spheroids. So I was wondering, how much of a role do the cells move at all, or when they divide, is there some kind of turnover with the cells on the outside to the inside? Does that complicate the analysis, or are they just little rocks and nothing happens?
So for the moment we perform immunostaining, so we didn't do live, but it will be very interesting to analyze what's going on live, because they are in this kind of size, they are not moving too much within the spheroids, but still they will divide, and so how it is and how nanoparticles can be internalized and then exocyte and go internalized again by other cells, and so this is interesting questions.
Yeah. No, for sure. Thank you.
And actually, I have a question for this nanoparticle. Since they are in the lysosomes, did you look at longer time point? If there is a degradation or any...
Not yet. No, we didn't go up to degradation, but usually these kind of nanoparticles, at some points, they will be degraded because they are really small. Yeah. But what is interesting is maybe not the lysosome part, but also the fact that we have some nanoparticles in mitochondria. So that might be interesting, even if it's a smaller part. That might be interesting for radiotherapy, for example.
And if you look at through your spheroid, in all the layers, the repartition between the different organelles is the same, or, for example, they are in different compartments, outside and inside the spheroid?
Yeah, for the moment we didn't, we quantify it globally, but it would be interesting to see if maybe at the periphery it's only in the lysosomes, and deeper it's in different regions. Yes, so we need to dig up and this.
Other question? Yeah.
Thank you, Charlotte, for this great talk. For the radiotherapy, have you tried to repeat the 2 gray fractions? And did you check if there was an oxidative stress ongoing or cell death ongoing beyond the cell growth of the spheroid?
Of the spheroid? No, for the moment we just analyze proliferation. This is the only output that we analyzed, but it will be interesting to analyze the ROS and to and also in situ, of course. But
and increase the rate to see if you can eradicate
the spheroid.
The spheroid.
Yeah, and we want also to go towards longer time also, because sometimes you have proliferation at the beginning, but then it's dropped down. So we want to go to longer time.
That would be nice.
I have another question.
Okay.
For the last part on the spheroid in compression,
Okay.
is there more cell migration due to linked with more stemness in the compressed organoid?
Well, for the moment, it was just we didn't look at the cell migration and we did not quantify cell migration, but it has been shown in other studies that when you compress the cells, you activate migration. So we didn't analyze at that point.
So, okay, if there is no more questions, thanks a lot for the presentation. So maybe we can do a test.
Can you hear me? Good morning. Can you see the screen and hear me?
Yes, we see well. You hear us well too? Perfect. Okay, so our second speaker is Francesco Pampaloni. He is a permanent scientist at the Buchmann Institute for Molecular Life Science in Frankfurt. Yes. So he is combining fluorescent light sheet microscopy and 3D cell culture to study cell physiology and tissue development. And so he was a coordinator of the European Consortium for Light Sheet Microscopy for Life Science. And he is a leader in the development of new technology for high throughput light sheet microscopy. So, and I think he will present some of this new development. So thanks for being with us through this broadcast.
Yeah, thank you very much for the kind introduction. And I'm really extremely pleased to be able to present our work to this really amazingly rich conference, and also online, but I was able to attend nearly all the talks by connecting with YouTube, so it worked quite well. So welcome everybody to my talks, and so I will present today some of new results concerning our technology that we developed in the last few years to achieve better, faster, and easier imaging, live imaging of organoids and spheroids. Right, first of all, I would like to just present you the people, the PhD students, very talented PhD students that make possible to achieve these results, and most of the data that I'm presenting today are the results of the work of these students. Essentially from two projects. One is over already in 2020, LSFM for Life, which was focused on the production of pancreas organoids for the cell therapy of diabetes type 1. The project was indeed very successful, and yes, we are seeking now to proceed with this project. And the second one is running. It's Brighter, which is focused on bioprinting now using light sheet based lithography. And as I said, most of the data that I'm presenting today are derived from the technology that we partially developed for both these projects. Okay, this is just to introduce the field of 3D cell culture. I don't need to give you an introduction about spheroids because they've been already beautifully described by Charlotte in the previous talk. And indeed, our work is mostly focused on organoids. So organoids at variance with spheroids. So spheroids are mostly compact structures which are filled with cells. At variance with spheroids, organoids can indeed have different morphologies that in part depend on the organ they have been derived from. So mostly we have epithelial organoids that derive from epithelial organs, and in most of the cases they have a cyst-like structure, so a monolayer, like a sphere, composed by monolayer of cells, and they have an internal lumen which is filled by a liquid which is produced by the cells themselves. But you can also have budding organoids and also branching organoids, so there are different morphologies possible. That of course depends on the culture condition and also on the organ the organoids have been derived from. Right. What are the advantages of using organoids? Well, essentially they are obtained by patient, directly by human subjects, patients, and they are composed by adult progenitor cells from the different organs. So you can have mammary gland organoids, liver organoids, pancreas organoids, and so on. And the advantage of this technology, I'm not going to go through all this list, but just to recall the background biology. The advantage is that these are primary cells, stem cells, as I said, derived from patients, but due to the protocol that has been developed in the past recent year by Hans Clevers and collaborators, and now there are many other former postdoc or PhD student from Hans Clevers that are really propagating this technology, including, for example, Marichel Uch, who is a collaborator of LSFM for Life. And yes, these organoids are primary tissues, but due to these protocols that have been developed, they can be cultured for a long period of time, not indefinitely, but for a long period of time, like a conventional cell culture, and they maintain an amazing genetic stability, remarkable longevity, and they can also be implanted in vivo, and so be very promising for the cell therapy of many types of diseases. Right, I have this last slide that gives you the background of our research. Also now in this talk, I'm focusing mostly on imaging issues. So in LSFM for Life, as I said, it was a quite successful project. So we were able to produce this long-term renewing human pancreatic organoids line and characterize them, sensing genetic stability, in vivo safety. We identified the optimal expansion and differentiation condition, and what was particularly important, we standardized the manufacture of these organoids under GMP production condition, which is of course an essential step to produce a therapy which can be used on patient, right? So this was very important. Our role in the consortium, so our group at Goethe University, we have been the coordinators, and our main role was also to characterize the organoids by using light microscopy, which is our specialty, so to say. And here we were trying to tackle many issues and challenges related to imaging this kind of cell culture. So we have a real agenda that we pursued in the last 5 years, and we are still working on it, essentially. And the main goal of this agenda, as microscopy is concerned, was really to streamline, simplifying, and standardizing the imaging of organoids in order to get the best possible scientific data, and that can be used to characterize the system. So we have very robust statistics, for example. And so what we need in order to achieve this goal was, of course, fast 3D imaging and live imaging, of course, as well. And very good to very good signal-to-noise ratio in order to be able to efficiently apply segmentation algorithms and other image processing algorithms, of course. And since we wanted to image the organoids live to follow the growth of the system live, we needed to have low phototoxicity. This is a very important requirements for live microscopy, of course. And as I said, in order to carry on this project, a standardized and simplified and straightforward sample preparation. And this issue of sample preparation is really the general topic of my talk. And yes, last but not least, a reasonable throughput. We are not talking now here of high throughput, but something reasonable. So to be able to rapidly exchange our specimen and start imaging, so something reasonable to get the amount of data that we need for the work, for the characterization. And yes, these being the requirements for microscopy, of course, our main candidate, which is also a focus of our lab, is light sheet microscopy in order to achieve these goals. I don't need to actually tell you about the principle of light sheet microscopy because I'm very sure everyone is acquainted with the functioning and work of light sheet microscopy. But yes, the important point which is the topic of this talk, is actually this part of the light sheet microscopy, namely the specimen sample preparation and mounting, right? So this is a very critical point. I have to say, for all type of microscopy, samples preparation is a central issue, a central challenge. So you have no matter how high-end, how cutting-edge your microscope is, if you don't have a very good sample preparation, then the data can be really poor, nevertheless. And so this is especially true for light sheet microscopy, and this is one reason that it was a major point of our efforts as technology is concerned in the past few years. Right, so this is again the geometry, the front view of light sheet microscopy and the top view. Yes, now if we are getting really to talk about sample preparation, in the last 15 years of light sheet microscopy development, there have been a lot of approaches to mount specimen, very creative ones, ranging from agarose columns, agarose beaker, other type of holders that are used for imaging embryos, glass capillaries that we used in the past to be able to apply clearing with aggressive agents like BAB, for example, which were working quite well, but still cumbersome and quite complicated to cut this glass capillary and to adapt them to the mounting system. And also much more exotic and strange way to clamp large tissues or hooks for entire mouse bones, for example. So we tried pretty everything. All this picture come from our lab. So as you see, over the years, we tried really everything, but what was lacking was kind of standardization. So a type of mounting system which was universal or relatively universal and suitable for all these type of specimens, and especially 3D cell cultures, which are a focus of the lab as well. So, and now in these slides, I'm actually showing you one of the results that we got in LSFM for Life for the imaging of the organoids, and then I will tell you how we achieved that. So what you see here is the container that we use, in which hundreds of organoids in matrigel, so cultured in matrigel, have been imaged for a long period of time, up to 1 week. And as you see here, in this kind of specimen preparation, we get two goals. We achieve two goals. So we have a lot of specimen inside one type of culture, one imaging run, and we are able to image them live over a long period of time, and with a resolution that allows us to determine single cellular processes, to a resolution which is good enough to observe the behavior of the organoids over time, looking at individual cells. So this is what we wanted to achieve. And so it turns out that in order to produce these containers, we found a material which allows us to build this kind of universal and easy to use mounting system. And this material, I'm sure that many of you already know it, are indeed microscopy grade plastic films, plastic foils that can be engineered in order to obtain containers of different shapes and fashions. There are several of these microscopy grade plastic foils available on the market which have different properties, particularly different refractive index that could be very useful, for example, for clearing. So you see this very high 1.52 refractive index foil, Xeonex, which of course is useful for clearing. But what we indeed used mostly in the project is fluorocarbon polymers. So one commercial name is Lumox, but in general you can call them FEP foils or FEP films, which have the very nice property of having a refractive index which is nearly exactly matching the refractive index of water. And then they are available on the market with a wide range of thicknesses, from very, very thin to medium thin and also thick ones. And what we used in all this, in our production, in our use, was a typical thickness of 50 micrometers. And then there are other type of foils available. The nice thing about these materials is that they can be shaped by using readily available techniques. Can be shaped to different type of containers, quite flexible way of producing these containers, which are suitable for imaging, for example. And as you see here, also by using these production methods that I'm describing in a minute, you're able to really shape the foil to forms which are to shapes which are very well defined. And this is very important for the technology. Okay, properties of these FEPs films very quickly. As I say, the material that we use is FEP, fluoroethylene polymer, or fluorocarbon polymers in general. The refractive index is very close to the one of water. Thickness around 50 micrometers was the best compromise that we found. And being fluoropolymers, so essentially Teflon, this foil is resistant to nearly all type of chemicals, even very aggressive organic clearing agent like BAB or acid or bases. It's actually resistant to everything. Very sturdy material. And it's once it's shaped, is also mechanically stable, also very thin, but mechanically stable. And due to the low thickness, is permeable to gases, so CO2, for example, and oxygen. It's not permeable to liquid, to the surrounding liquid media. So the advantage is that in your container, you will have a media composition with it, which is constant. And also a very tiny volume, which is handy when you're using expensive media employed in the culture of stem cells, for example. But they are permeable to gases, so you can easily fine-tune the gas composition of the outside and to adapt them to your culture condition. This is nice. As I say, they are tight, impermeable to liquids, small volume, and given the sturdiness of this material, they are also autoclavable at 120 degrees and 2 bar of pressure, so like typical autoclave condition. We used quite early this kind of material in the past. I was still at the MBL at the time and working with Philip Keller. We were determining the mechanical properties of microtubule in the microtubule aster in a Xenopus levis egg extract, so a nearly physiological environment for the microtubules. And we did all this measurement with light sheet by using also at that time a FEP foil container, which was quite primitive, was efficient because, as you see, we were able to really get nice data out of it. But as you can appreciate, maybe it involved a lot of gluing and adapting to glass capillary, so it was not very handy producing these bags. So, and this is what we tried, starting from this early experience, we tried to optimize this approach. And this is what we actually produced. Essentially, the most straightforward way to think about that are cuvettes. So, like mini wells in which you can pipette your specimen from the top. So, in a very similar way to what we do for multi-well plates. So, this was the goal, the idea in order to make the process easier, as easy and straightforward as possible. Right. So this is what we realized in LSFM for Life. So these two type of cuvettes, one which was adapted to our MDSLM, which has a stage which is below the specimen plane, so to say, and we did another type of cuvettes which was optimized for live imaging and optimized for the light sheet setup implemented in the size Z1 microscope. So, and I'm going to talk about these two types of cuvettes in what we've got during LSFM for Life, and then in the third part of my talk, or second part of my talk, I will tell you about our unpublished results concerning an exchangeable specimen chamber. So, but I'm focusing on that. We also did the third thing, which was cuvette or plates for light sheet microscopy in inverted geometry, but I'm not focusing on this topic today for reason of time, and we are still finishing this work, and I hope that we'll be ready next year to publish also this part. Right, how to produce this shape that we need in order to produce a cuvette, for example. And the process that we use is very well known in the industry, and it is thermoforming or vacuum forming. Essentially, if you have a foil, you can create any type of shape out of it if you heat it up, and then once it gets close to the melting temperature, you actually press this foil onto a mold, a positive mold, for example, and you activate a vacuum so that the foil is really pulled down and adheres perfectly to the positive molds. This is something that is used ubiquitously in the industry to produce any type of stuff from anything, essentially. And we adapted this method to the laboratory bench by using a quite cheap but effective instrument that I'm showing to you in a second. So this is a video. I don't know, I don't know whether you will hear the audio, but it's not important. It's just 1 minute. As you see here, this is this very simple machine in which you have a heater on the top, the foil here, and the positive mold here. Now I'm giving a final kick of heating with an heating gun, as you see here, so a stream of very hot air. And then this allows us to reach nearly the melting temperature of this Teflon-like material. So now you see that the temperature is going to be reached. And in a second, I'm activating the vacuum and pulling down the foil on the shape. So, yes. So, and now it's finished. So what you see here, this is the final result. Here you see the mold, one of the possible mold, which is a cuvette indeed connected with a glass capillary. And you see that after thermoforming, this positive shape, this positive mold has been perfectly formed, transferred to the foil. And this allows us to produce a lot of containers, as really we need and we wish. We can produce, we are very flexible, and we can produce different sizes that adapt to different sample sizes, right? And also different shapes, from rectangular or square cross-section to the pyramidal or trapezoidal that we need for the inverted light sheet microscope for this kind of plates. Here you see we produce an array of these cuvettes, then we cut them with scissors very easily, and we can mount them to the holder, which is then mechanically connected to the light sheet microscope stage. Right, this you see here the cuvette. Here on this picture, you see the empty one, which has been mounted essentially glued very quickly on the pin, which is connected to the stage, as I said. And then in this kind of containers, you can easily pipette whatever you want to image with a light sheet microscope. This can be spheroids or organoids. You can have a liquid medium, or you can have an hydrogel like the Metrigel or collagen, as you wish. So as you see, this was meeting the requirement that were set at the beginning of having a nearly universal type of container, and easy to handle because you can simply pipe it. Here in the inset, you see an example of two large organoids inside. And these are the results that we published in nearly 2 years ago already. Firstly, by looking at the use of this cuvette to image fixed human pancreatic organoids, but also tissues, like in this case mouse brain or mouse ovaries, so ranging from a small three-dimensional sample to large tissues. This was possible by using this type of containers. And so we demonstrated that this, we called then ultra-thin fat cuvettes, that we demonstrated that were suitable to image specimens ranging from micro to millimeter size. Right. And since this cuvette can be very tiny, then you can also easily apply indeed multi-view imaging in the light sheet microscope. You can easily rotate the entire specimen, and in the case of organoids, by recording 4 views, it was possible to fuse them using well-known algorithms and obtain an isotropic imaging of this large specimen by using the cuvettes. Okay, now second goal was really to demonstrate that this system are suitable for live, long-term live imaging of the organoids, like I'm showing here in this video that we eventually collected the system. So you have, as I anticipated at the beginning, you have hundreds of organoids in your cuvette, and you want to resolve the behavior of even individual cells, of individual organoids, but also individual cells inside organoids. And with light sheet microscopy, we can do that, and the point was really to increase the throughput. Of course, as I said, not high throughput, but having a reasonable throughput and making the process streamlined and straightforward. What we wanted to achieve was really to have long-term light imaging and a multi-scale analysis of the organoids. And we devised for that a pipeline, including incorporating a light sheet to observe the organoids at the micro and meso scale, and finally coupled with another pipeline involving essentially very simply bright field in light imaging in bright field, in which we are able to ascertain parameters relating to the behavior of entire culture, so the macro scale. And so this pipeline was so ranging from high content type of imaging with a reasonable throughput to really high throughput assays, as you see at different multi-level of spatial resolution. But first of all, we wanted to demonstrate that the cuvettes were suitable for long time culture over the organoids without inducing any change or influence onto the phenotype of the growing organoids. So this we first demonstrated just in the incubator by setting pipetting organoids in the cuvettes and then looking at them over time in bright field microscopy. So, and as you see here from this video, the fat foil, this condition, and the culturing into these very tiny chambers was not influencing at all the behavior of organoids, which were just growing normally. So this was very important to characterize. So at the end, we concluded that the fat cuvettes are suitable for direct seeding and culture of organoids, and they can culture in the cuvette for a period of 10 days or even more without influencing the behavior. And this is then what we adapted for the live imaging with the commercial size light sheet microscope. As you see, in this case, the setup has the stage from the top, so you have a cuvette that actually arrives the imaging plane from the top. Yes, but we produce it exactly the same way by using our positive mode. And here you see a close-up of the cube. And yes, by using that, we were able to achieve this long-term live imaging, as you see here over a large population of organoids and looking at their behavior over time. In this case, you appreciate this rich diversity of organoid behavior in the culture. So you see not only human expansion, but you see cell division, and then you have the rotation of the organoids inside the matrix, and even the migration of entire organoids. So it's a very diverse, heterogeneous behavior that we wanted to characterize. And the starting point, of course, in order to do that, is having very good microscopy data. Eventually, you have, after 10 days, you have the death of the organoids, which is an entire life cycle, so to say. And as I said, these data are very, in the light sheet microscope, you have usually a quite good signal-to-noise ratio, and this made possible to apply segmentation algorithms to the organoids and obtain quantitative data out of it. And here you see an example of the segmented cultures. More than that, we wanted really to look at single organoids, a single cell, in order to characterize all these different aspects of or different phenotypes in the culture. And so over time, we looked at the formation of the organoids, starting from individual cell or clump of cells. As you see here, we looked at the polarization, so the formation from this aggregate, then the formation of the hololumen and the polarization of the cells facing the lumen, like here. Size oscillation of the organoids, which was, it is a particularly intriguing aspect of organoids dynamics and growing. That the organoids have a lumen, and this lumen is actually interacting with the outside environment, and you have a pumping behavior of the cell in many cases that induce a rhythmic contraction of the organoids as a whole. And characterizing this contraction is a useful assay for example, it has been used for characterization of this organoids obtained from cystic fibrosis patients. And yeah, the pumping behavior is strictly related to the functioning of the ion channels, which are then undergoing a mutation that eventually induce cystic fibrosis. So an assay characterizing the pulsing of the organoids is indeed useful to screen for drugs for this for cystic fibrosis, as it has been done and published by the Clevers group, for example. So we wanted to characterize that, but and also going at single cell level, the mitosis, cell division, and then again back to macro scale, the fusion of various or multiple organoids to make a large one. And then the luminal dynamics, so the formation of multiple lumens, for example, in the organoids. And as I said, also to characterize and model the rotation of entire organoids inside the three-dimensional matrix. And here, so all this characterization we published this year, early this year. So I'm not actually going through all this kind of data or models that we did, but essentially showing you some glimpse, since I would rather like to focus on microscopy this talk. So as you see here, the quantification of several parameters like the number of cell nuclei for 3 types of spheres that you can encounter in the culture. So a small one, medium sized one, for example, and a large sphere. So you have all this type of heterogeneity in the culture. And for all these 3 types or classification of organoids, we measured these phenotypic parameters or morphological parameters, like the number of cell nuclei, the volume of the organoids, the surface area ratio, the mean number of cell nuclei in the neighborhood, another morphological parameter, or the mean density, and so on. So this was possible because of the 2 aspects of the microscopy, the long-term live imaging, so low phototoxicity given by the light sheet microscope, and the eye signal-to-noise ratio. Okay, this is another example looking at the volume analysis and this pulsing behavior. And so overall, in this work performed in the main framework of LSFM for Life, we were able to have this achieve a multi-scale analysis of the organoids, combining microscale, mesoscale, and macroscale analysis in this pipeline, and looking at all the different aspects that I went through. Okay, so in the last 10 minutes or less, I'm showing you, so this is the final slides of this part. So to summarize what has been achieved, so the fast 3D imaging, then long-term light imaging, and easy and quick specimen preparation, which allows us to get an increased throughput. Okay, so, and yeah, last aspect, no invasive mounting such as embedding in agarose, because the organoids are pretty fragile structures. So if we try to embed them in agarose at the beginning of the project, and we realized it was really heavily influencing the dynamics, of course. So as I said, in the next 10 minutes or so of the talk, I'm describing you now unpublished results, which have been indeed obtained. It's a work that I mainly perform in collaboration with Julian Colombelli in Barcelona, at IRB in Barcelona. So we are working on it I've been working to it for nearly 2 years now, and we are getting nice results. All again along this philosophy or this strategy to make light shift microscopy imaging easier from the point of view of specimen preparation. So it's not a drastic revolution in imaging or a new type of microscope, but it's really trying to make life easier and for research indeed. So. So here, if I go back to my experience by using light shift microscopy over the last 10 years or so, I would say that the specimen chamber is still was and still is a limiting factor in light shift microscopy, something very cumbersome and really, for the user, it's quite troublesome. So you might have with conventional chambers, as you see here, which involves a lot of screws and tubing, especially for light imaging and connection, and they are quite bulky as well. So you have many issues, many frustrating issues, essentially leaking windows, leaking O-rings, screws, more screws, and fittings. And then another point is that in this type of chambers, the objective lenses, which are typically water-dipping objective lenses, are in direct contact with medium origin index matching solution. And so this produces several issues like corrosion, etching, specimen contamination. And another important annoying issue is that you are not really able to detach the chamber from the microscope if you want to put it back in the incubator or make some changes under the sterile hood and so on. So you cannot do that because you would have to detach the entire objective lens and carry it on to the other bench, which indeed I did in previous works, and it was a nightmare. So starting from this bad experience, we try to improve that. And as I said, we now develop what we are calling a dry immersion technology. And in this case, a dry immersion LSFM chamber for multiple or serial imaging of three-dimensional cell culture. As I said, quite exciting work or funny work that I'm doing with Julian Colombelli, and we started during my sabbatical in Barcelona last year, 2020, also under quite difficult conditions to the pandemic. So the idea now, what is the idea? Is that, so we take advantage again of the fat foil, mostly, but other foils are possible. But what we do now, let's say we have a detection objective lens of the light shift microscope, and now we do not put it directly in contact with the surrounding medium, but we use the foil as an intermediate bag to contain our real specimen. And this container, which is flexible, resilient, right, can be put tightly in contact with objective lens, making at this point a very thin optical window that allows you to use for example, water dipping objective lens, because we don't have to forget that the refractive index of the foil is close to the one of water, and moreover, the foil is very, very thin. So even if you have an index mismatch, this would be quite negligible concerning in deteriorating the point square function, for example. So by using this strategy, we are able to make items that or sample containers, which can be the LSFM chamber, but even plates, so normal multi-well plates, in which the immersion medium is not in direct contact with objective lens, and so we have a kind of dry immersion. So this is the general idea that we are pursuing. Now let's go to the chamber, to the spin chamber. By using these two aspects of the technology, so on one type, rapid rapid prototyping with 3D printing, and vacuum thermoforming with this small machine. On one side, we produce positive molds with the shape of the immersion objective lens, and over these molds, we formed our foil in such a way that here in the middle and in contact with the front lens of the objective, you have a very nice, optically clear and neatly defectless optical window, very thin, that gets in close contact without an air gap, the objective lens, and allows you to produce now a chamber in which the objective lens is not fit inside, but is just put in contact with the window. So you have a self-contained chamber that you can remove and put back, remove and put back. So this was the idea that we eventually realized. Of course, now for making this chamber, we do not rely on the mechanical workshop anymore, like previously, but we can use 3D printing to make a monolithic chamber in which it has been designed in such a way that you don't mount all the parts, like the windows or even these fat foil windows with screw or O-rings or other types of complicated and shaky fitting, but actually make a block, a monolithic system in which you insert the glass window at the side, a fat foil panoramic window on these sides, and the internal optical window on these sides, and the connection with the stage on the bottom. You insert all that by using cavities, and then you seal all these cavities by pouring a biocompatible silicone gel, which is in a discovery of Julian Colombelli, and it's quite universal blob that we use in the lab. It's this green blob, which has amazing properties, and is able, the important thing, is able to seal all your chamber in one step, and it's a very sturdy type of sealing that doesn't leak anymore. A nice thing is not unremovable, so once in a while you want to change it, you can just remove it with forceps and pour it again. So it's quite practical way to do that. And here you see, yes, the detail of this chamber and these various cavities, for fitting also tubing and temperature sensor for live imaging. And from this side, you can clearly see that you insert, in this case, you simply insert your water dipping lens into the chamber, and it's facing not the liquid inside, but just an optical window. And yes, serial imaging implies that you can for example, use a chamber that you have in the incubator, you take it out of your incubator and you insert in the microscope, and you perform your measure, and then you remove it and you put it back, and you can get another chamber if you wish. And in order to realize that, we design a system based on magnets, in which actually the docking is very fast also in this case. We just have, as you see, 4 powerful magnets here on the stage, and 4 powerful magnets on the chamber below, and then we make a simple click and we insert the chamber, and we can secure it with 2 screws, but essentially it's a very simple one move that allows us to insert the chamber. How does the chamber perform optically? So this was the next thing, and it performs quite well. Here you see an example of spheroids in water in this case. You see the procedure. You have your beaker here. You pipette 1 single spheroid or multiple spheroid in it. You insert in the chamber, and you image. And you see that in water, the optical quality was pretty good. Of course, as you see here, the spheres are not cleared, and so the penetration depth, as we heard in the previous talk, is limited to some to certain layers, and not even to the half, right? But this is a common issue, of course. So this is also what Julien did in his lab. Julien and his group, we are working indeed in parallel in developing that. And Julien also implemented the same system on an axially scanned light shift microscope that they built in the lab. And you see here, in this case, imaging organoids, you see that the optical quality is really very good by using this principle of dry immersion. So, and also importantly, this ability, the fact that the objective is not inserted directly in the chamber, but just pressed against these optical windows, allows you something that was previously impossible with the conventional light-shift microscopy, namely change objective lenses with a turret, as it is done in conventional microscopy, inverted microscope, for example. So you see here that in this case, for this video, Julien was using a 5x detection lens in air, and then he switched objective to a 25 high-resolution lens in the same specimen without removing anything, yeah, just switching an objective in a turret, and you see the results, which are quite nice indeed. And what about the point spread function? Does the fat foil deteriorate it? So here you see on the left the contact of the front lens with the fat foil. So I hope you can distinguish it. This is essentially the front lens. And this is the point spread function without foil and with foil. And as you see, the result is that the degradation of the point spread function is very minor. Finally, so all these measures have been conducted in water medium, which is nice also for live imaging, for example. But is it possible to also use, as it is, the fat foil for clear specimen? And the answer is yes. So we tested this on organotypic brain slice culture, in which you have a biopsy, or not a biopsy, essentially from a brain tissue from mouse. On this tissue, you can put a brain cancer spheroids and look at the spreading of the cells, right? This was an assay that we did. And in order to determine the spreading of the cells, for example, we use cubic 2 as a clearing agent. And as you see here in the cuvette, we have our specimen, the organotypic tissue culture in cubic, and we image that. And the result is that, yes, we are this type of chamber as it is, without changing, switching from fat foil to another type of foil, is thin enough that you can image with cubic 2, for example, without loss without degradation of the image. And as you see, here we have a nice signal-to-noise ratio, again, that allows us to segment the specimen. Finally, live imaging. So this was also a very important thing that we want to use increasingly, live imaging, of course. And so in with the Z1 size microscope was nice and working, but not really essentially fast. So we want to increase the throughput in this case. So, and the way is using this removable chamber. Here, as you see, we adapted the chamber for live imaging, inserting a very easy way for the tubing. So you see the inlet, the outlet, and the temperature sensor here. And here, the nice thing is that you don't have a culture media directly in the chamber. Here, what you have here around the specimen, which is in the middle, in the cuvette, you have just a bicarbonate buffer to ensure gas exchange with CO2 and oxygen. But the real specimen with the medium is inside the cuvette, and is also sealed with the green blob, so that it's completely isolated from the rest. So you don't have really to have concerns about long-term sterility of the specimen, right? And here we are implementing a system which was published a year ago, in which the medium, or in this case the buffer, is circulating about in a very long silicone tube, allowing which works as a gas exchanger. And then, as you see here, the silicone tube is in a small incubator, and then the peristaltic pump is circulating here in a closed circuit here, circulating the buffer inside the chamber. And inside this cavity here, you have a knitting foil that keeps the temperature constantly very reliable at 37 degrees. Finally, very last slide. So what Julian and this group are working on is extending this technology to not only really limited to light sheet chambers, but to multi-well plates. In fact, with multi-well plates, we have really the same problem when you're using immersion lenses. That the immersion liquid evaporate in the case of water over time or very quickly possibly. And if you want to have multi-well imaging, so high throughput imaging of your specimen, actually you have to replenish the medium, otherwise you will lose it. And so there are, of course, ways to replenish constantly the immersion medium, but they are quite technically quite challenging and sometimes not really reliable. So what we thought it was to indeed adapt this foil technology to make an intermediate layer in which the immersion medium is not outside but is contained by the FEP foil, and the objective lens is in tight contact with it. And it seems to work very well, and so it's leading us towards a very long-term imaging without refilling. And you don't have evaporation, no leaking, no cleaning, and you can also swap the objective lens easily to have multiple resolution and magnifications. Right, this is the wrap-up, but I'm thinking I'm getting out of time. I think I told you the most important thing concerning this dry immersion technology. So a final remark, as I say, this dry immersion technology is not only applicable to LSFM, but also to inverted microscope, and this can open new possibilities. With that, I'm at the end of my talk, and I would like to thank, of course, all the collaborators in my group and at the MLS, and Julian Colombelli, of course, at ERB Barcelona, and the other groups that collaborated with us over the years in the framework of LSFM for Life and the Brighter project, and the funding agency, the European Union, and the German Ministry for Research and Education. And I also thank you for your attention, and I will be very pleased to answer your questions. Thank you very much.
Oh, so thank you. Is there questions?
Yeah, thank you, Francesco. Beautiful talk, as usual, with very nice movies and images. I have a question about actually the robustness of the FEP foil in terms of being able to preserve the shape. For example, what about if in your cuvette the sample is growing like a lot? Are there any volume variation, and does it affect the flatness of the surface of the cuvette? And this can be extended as well to the last situation you've shown, if the FEP is actually deformable, and what are the consequences on the optical properties and the flatness of the surface?
Right, very good question. Essentially, the cuvette is mechanically stable, but of course it's a thin foil. So if you actually touch it with forceps, so with a certain force, it will deform. But it is resilient on one side, first point. So it comes back to the original shape. And second point is that it's not, in the case of water immersion, is not really crucial that the cuvette has a perfect shape. So maybe you have appreciated that this perfect square shape sometimes is not there in the imaging, but this is not really relevant, because since you have the same refractive index compared to water, then this will actually shape changes or a more irregular shape of the surface are actually irrelevant for the imaging in case of immersion in water. Of course, if you are putting your cuvettes in clearing media, like for example cubic 2, then this is more critical, and you have to take into account that you need a shape which is quite well defined and not deformed. So this requires more careful quality control when you produce the cuvettes by thermoforming, and the more careful handling. but this is clear. But for imaging in water and so in PFA, in fixed aqueous media, or for light imaging in aqueous media, is not a big issue, the size of the cube. Concerning the optical window, yes, this is of course an issue. You have to really make sure, since now you have a huge mismatch essentially, because the water dipping lens is not directly in water, but there can be potentially a layer of a gap of air, and then again the foil and the water. And if you don't have a very good connection or adherence of the foil to the front lens of the objective, then you have a gap of air, and then you will have a mismatch that will inevitably deteriorate your imaging. There, Julien, we devised some tips and tricks to ensure this strong adherence. Of course, you can push the objective lens to the foil in order to ensure that. Although a strong pushing is not recommendable because it can introduce some lateral strain, and this strain also makes some irregular contact. But we devised some tricks that hopefully we are going to publish them soon. But yeah, you try to remove the air and to make a kind of really a tight adhesion with that. But we have multiple ideas to tackle that. Of course, yeah, for implementing this type of window in the light sheet and also in the multi-well solution, you really have to ensure a close, a very nice surface and a close contact. Yes.
Hi, Francesco. This is Lydia. I to follow up on the question of Gaël, I had a question concerning the geometry of the FEP, because most of the time you had let's say a circular shape. And I was wondering, is it really transparent, or do you have to optimize the orthogonality to the light sheet? So how do you manage that?
Right, so the FEP foil is completely transparent, but of course depend a little bit also on the thickness. So if you are using a thickness of 50 micrometers or less, there is no transparency problem. It's really completely transparent to the wavelength, and you have no image deterioration during to that. But by increasing the thickness, especially over 100 micrometer, we'll start noticing that can maybe absorb some light, or the transparency is a little bit less. I mean, there is also a lot of experience by using the FEP foil for developmental biology, especially Jan Huisken has been establishing also this method by using tubes which are slightly thicker. In my experience, this leads to a decreased transparency, not necessarily a decrease in image quality in terms of degradation of the point spread function, but a certain decrease of signal. Yeah, this is at least my experience with this material. Yeah.
I have a question for the thermal forming of the optical window. Which kind of mold do you use to have this optical transparency? Is with 3D printing is working, or?
So the question is about the mold, how to produce the mold for the formation of the windows.
Yeah, especially for the optical transparent windows, because I know with 3D printing it's sometimes it's complicated to have a transparent, a nice transparent surface.
Yeah, so we use, in order to prototype the positive molds, we are using 3D printing with stereolithographic 3D printing, so that allows a very nice resolution. But yes, this is a very important point. We need a very smooth surface for the front lens to properly have a high quality front lens, and there we find out some tricks to post-process the 3D printed molds in order to have a very smooth surface. For example, I can mention one trick which has been invented by Julien also, is that you have your ready 3D molds. Let's imagine the one with the objective lens, so that have the flat top, which is actually the part in contact with the front lens of the objective lens. And yes, what you do after printing, you deposit a very small droplets of the liquid resin, which is used in the 3D printer, and then you let them spread, and then you place over this droplet a very essentially glass cover slip, which has a very well controlled, flat and smooth surface. And then you illuminate with 4 or 5 lights with an LED lamp that you use to cure this resin. And then at the end, the resin hardens, and you detach the cover slip. And below, what you have then on the mold is a very, very perfect smooth surface. And then use that to thermoform the foil. And in this case, the surface of the foil is very smooth as well, and the quality is very high. Although all this stuff is produced in the lab. Okay, so it's something completely not industrial quality control levels. It's something that we do on our bench. So if we imagine then that in the future this kind of items can be produced by company using more stringent and quality control and better optimized technology, then here you can really have quite kind of mass production of these items in high quality. Yes.
Okay,
but we are pretty happy as they perform now, even by using this. Production on the bench in the lab.
Yeah, thank you, Francesco. A question about the long-term survival of your cells. I was really amazed to see them growing for 10 days. So if I got well, there is gas exchange through the fab foil, but then you don't refresh the medium. So you have to think about, I don't know, how to supply nutrients enough for 10 days. How do you cope with this challenge?
Yes, this is also a very good question. Indeed, this was a problem for the Z1 system. Replenishing of the medium was not that easy. At the end, you see, after a few days, I don't remember, it was 7 days or like that, the organoids start dying. This is the natural cycle, but also due to the fact that we weren't able to really efficiently replenish the medium inside the cuvette. So this was a problem. And indeed, this is now what I hope will be solved or circumvented by using the exchangeable chamber, because this is exactly one of the advantages that we hope is going to work well. So essentially you image for 3 days, and then you can stop temporarily your imaging, you remove the chamber, then you go in the under the sterile bench, you pipette, you exchange the medium in the cuvette, and then you go back to your microscope and you start imaging again. This is one way. Otherwise, another solution would be by replenishing the medium with a very tiny capillary that you can insert in the chamber. But we also tried it, and it can work, but we always had some kind of contamination at the end, or formation of tiny bubbles that were disturbing the imaging. So I hope that indeed it will be easier by using the removable chamber. Of course, it might be difficult to find exactly the same position, but yes, since we are indeed imaging the entire cuvette nearly, you will have a displacement of the specimen, but you should be able to register it back once you have imaged all the time, and be able to find back the region of interest that you wanted to observe.
Okay, thank you.
Thank you very much. Nice talk. I have a question concerning live imaging. How do you deal with the fact that for in-depth analysis of your cells, are you able to segment your cells up to the center of your organoids? How do you deal with that?
So the audio was a bit shaky, but maybe I understood correctly that how was the imaging inside the organoids?
Yes.
Or in Z?
Yes, in Z, yes.
The organ. This is also a good point. So the organoids are, from a point of view of a microscopist, are quite nice. At least this type of organoids, because they are essentially a all of monolayer, right? So you don't have a lot of cell layers that you have to penetrate in order to image the cells more inside. Of course, you have also organoids that get field of cells. It depends. For example, tumor organoids can also be full, like spheroids. But in general, at least the pancreas organoids were typically monolayers, all of monolayers. So it was not an issue. Penetrate in Z, going very deep in Z through the sample and imaging all the single organoids, simply because they are all of inside, so you can penetrate easily through them. Yeah. Of course, having spheroids or completely or compact organoids then would be an issue, the same issue that you have for all the specimens which are not cleared, so that you have limited penetration depth. That issue that you can circumvent to some extent by rotating the specimen and performing multi-angle, multi-view imaging. Yes, so this is possible. But indeed, the organoids, as we had, from a point of view of the microscopy, were quite nice specimen, yes.
Okay, so thanks a lot. It will be the end of the session, so people can have a small break before the next talk. But thanks a lot for your very nice presentation.
Thank you very much.